for today's ceremony.
Thank you.
Taking the ceremony forward, I would like to introduce Professor Dr. Arun Jamkar, Distinguished Professor SIU, ex-Vice Chancellor of Maharashtra University of Health Sciences, Nashik. Dr. Arun Jamkar is the Chief Medical Officer in Index Technology and Senior Consultant in Persistent Systems. Sir is the Director Academics at Galaxy Care Hospital, Pune. He's thought leader in pods.com and mentor and board of management in rises.com. Sir has spent 3 decades teaching surgery and performing thousands of major operations in places like Grand Medical College, Mumbai and BJ Medical College, Pune. His consistent inclination towards medical research resulted in a PhD in surgical oncology from National Institute of Virology, Pune. A significant milestone of his journey was attaining the position of Dean of BJ Medical College, Pune. Sir was also a part of managing the swine flu epidemic and disaster management of German bakery bomb blast. Sir has helped create an ecosystem for interdisciplinary research between science, technology, Ayurvedic, and homeopathic medicine. One such example of interdisciplinary collaboration is clinical engineering by creating engineers for hospital. Sir created a movement of teaching bioethics and medical curriculum through National Nodal Center of UNESCO Chair of Bioethics in Haifa and all state medical universities of India. Sir has 64 published papers in various national and international journals. He has received various fellowships such as FICS, Fellow of International College of Surgeons, FMAS, Fellow of Minimum Access Surgeons of India, FIAGES, Fellow of Indian Association of Gastrointestinal Endosurgeons, FIAMER, Fellow of Foundation for Advancement of International Medical Education and Research Institute. Sir is joining from US today. We thank you, sir, for joining us at this hour. He will be delivering a session on research methodology for medicine today. I welcome you, sir, and request you to please address the gathering.
Sorry, Adori, if I could add.
Yes, sir.
He is Distinguished Professor at SIU, and I'm proud to have him as a member of our family at the Symbiosis International University. So please mention that.
Sure, sir, yes.
Are you seeing my screen?
No, sir.
Okay.
Yes. Yes, sir.
Okay. Honorable Chancellor, Professor S.B. Muzumdar, Dr. Rajiv Eravadekar, Dr. Vidya Eravadekar, both are my students, Dr. Anand Bang, Dr. Yadnik, and obviously today's ceremony belongs to Dr. Sharabari Shukla. So let me first thank Dr. Sharabari Shukla for inviting me to give a talk on research methodology. Obviously, this is a little different, where I am going to talk not only of methodology, but how methodology was taught to me, and my failures have make it perfect. Let me first give compliments and congratulations to Dr. Shukla for starting the statistical clinic. As a researcher, having such clinic is almost music to my ears. And I hope you'll be just flooded with requests. Dr. Rajiv Eravadekar, I'm going to request you that all institutes across Symbiosis, we should have a, what you call as a technical committee to evaluate all research projects, including our medical college. And therefore, this technical committee has to be have a de novo by secretary or by some invited member, a statistician, because the commonest cause of failure to get our projects say published is statistical design. And this is some blind spot, as you rightly said. So I think we need to have such technical review committees across all the institutes and with a statistician in charge for advising us on statistical background. I'm just going to start my lecture by giving my tributes to my two undergraduate and postgraduate teacher, Dr. Vijay Gharpure, who was my professor when I did my MS from Government Medical College, Aurangabad. And he was guiding to my dissertation. And believe me, my dissertation for MS general surgery was immunosensitized porcine lymph node cell infusion followed by a xenogenic transfer factor in carcinoma maxilla. So this is supposed to be maybe a grandfather of today's monoclonal antibodies, and I'd work on immunotherapy of cancer. And there are, say 2 years before, when we got a Nobel Prize for this, suddenly everybody started paying attention, and this is now we called as an immuno-oncology. But then my basic, flight of research was given to me by when I was an undergraduate by Dr. M.B. Gharpure, who was my professor of pharmacology. I worked him for several times, and I worked with him for what is called the PA values. So an acetylcholine atropine antagonism, at certain stage, it becomes non-competitive to competitive. And then I, as an undergraduate, helped him to find out that point. And then he published, and I don't have his picture because it was too old, but then he gave me that publication with best compliments because I contributed to his, what you call it as research. Then he gave me an independent research on what is called antihistamine resistance in Lucian grass eating rabbits. And the first lesson I got in the control group and the study group, there was so much of statistical variation, more than 1,000 times, 1,000%. So that whole process became irrelevant. So I think that was my first, lesson from statistics. Then when I came to Pune as a professor of surgery, I did my PhD under Dr. S.N. Ghosh, who was a director of immunology in the NIV. And my PhD thesis was on NK cell interferon system. And we are first to start and think of considering, let's say, interferon clinical trial in oral cancer, which is supposed to be because of a viral origin. And then obviously all my clinical side was guided me, and I was inspired by Professor Dr. Mrs. Mehta. I think these are the four individuals on whose shoulders I think I had been finding out that. Now my topic today, I'm just going to have maybe in earlier 15 minutes of discussion on say research as a philosophy and university, and then we'll go as a research as a process. And subsequently, I just want a lot of things to be done at what is called as self-directed learning. And therefore, I'm just going to just sensitize them about the process. Now, the research has been given a lot of importance, and I think we need to cultivate a research culture. I'm very happy Symbiosis University is doing a lot of things to develop and a culture of research, because it is taken as indicator of excellence. And a society becomes more knowledge intensive. It becomes more and more dependent on such institutions such as universities, where Symbiosis is playing a role to create a knowledge, educate people, provide people with learning processes, and promote research and innovation. I think that is one thing which is there, because we need to understand that today's world, the data is there, but then data is generated by knowledge. So knowledge is economy. The new roles of today's universities emerge from the development of a new world economy, wherein knowledge, creativity, and innovation play an ever-increasing pressure in generating and sustaining growth. The society has entrusted university with immense responsibilities to endeavor human development. Governments increasingly turn to universities for solutions facing problems, and industry also turns to universities for cutting-edge knowledge needed for the patents, products, and services. So at that whole process is now going towards research. So what is required is to cultivate a whole, phenomena of research, because research in any area requires continuous intellectual engagement. Very important, continuous intellectual engagement. And in addition, we should have what's called passion for idea and creative thinking. So these three combinations of, let's say, one of the virtues would create a better area of research. And then what are the things that research can do? It answers scientific questions, it solves problems. And basically, all research has to be directed to improve the quality of life of a common man. It may be anything. It might be health, it might be technology, it might be any aspect of life. But then that quality of life has to improve. And therefore, it investigates important questions, connects prior study to research, and uses appropriate designs and disseminate results. So research has to be creative, strategic, original, imaginative, and then you have to think out of box. It should be, need to be fluid, flexible, and be inspired, imaginative. So these are all qualities that become creative. And there's nothing like a best research, and there need to be good questions. And don't people all the time say that we don't have ecosystem. I think my research project of immunotherapy can show that in a small college like Government Medical College, Aurangabad, that whole thing was done first time in the world. So we are basically two types of research: the basic research, what is called the problem-based research, market-driven research. And then what is required is we need to have a combination of a fundamental research and inspired research. So the classical example of fundamental research is Bohr's, or maybe classical example of inspired research in Edison's bulb, whereas the combination is a pastures. And say teachers and faculty, they go on complaining that they don't have time. But I think research should always have time, because there is an academic space where it can create into ideal university. Now, coming back, the whole research has to be driven towards creating some products, creating some say, intellectual contribution. And therefore, the whole thing that we need to think of business, and the university and business, and university and industry. And then we have what is called as an academic community and a commercial world with the SMEs. And therefore, we need to have a transfer of knowledge across, and therefore we can have the RD funding. I think this has to be done. Now, earlier, universities were not doing any research at all. And then first time, what you called as the Bay-Doles put a thing in Senate. And then from that, the American universities started giving more attention to research. And therefore, the growth in patenting started being more and more after these Bay-Doles criteria. And I think that is what is required, is that the whole thing has to go up, and then create a system by which we, as a university, do more and more basic research, because this is pressure going on the basic research, and because basic research takes a long time, but then it has a long-term impact. Therefore, whereas a market-driven research has a short time, but then we need to have a combination of a, say, Malaysian model, where the Malaysian model, they want to say that 2/3 thing has to be market-driven research, whereas 1/3 thing has to be basic research. Therefore, this combination of these, both of them together, need to do, say for better humanity. We need to think of, as now in medicine, because industry-sponsored research, where we are, a lot of things can be done in manufacturing, industry, and the pharmaceutical industry. Therefore, what is required is to increase that knowledge base, which has, because of the content of the processes, practices, and then technological capability has to be increased. And therefore, once we do that, the faster knowledge can be turned into products, because that is more important, is what's called the rapid prototyping. And therefore, we need to understand that all this background has made a university more responsible. University has a role to play in translating research to knowledge and disseminating to conduct further research, because this, I think, whole responsibility given to all, to the universities. And at the same time, we need to think beyond what is called as innovations. So research converts money into knowledge, whereas innovation converts knowledge to money. Therefore, whatever knowledge we have, first we do research, get innovations, and that those products will give payback to money. Therefore, this is a circle. Therefore, we need to think beyond and create more innovations. And I think these are the areas where we can do innovations. Now, Symbiosis being one of the ideal progress university, we need to think in this term. So creating either industrial parks, ICTs, technology parks, incubation facilities, and where it could be funded either by industry, university, and then actually, of course, there are a lot of, say, funding coming from Government of India, like BIRAC. And therefore, what is required is, once we have get into collaboration, we need to think that the scientist working in industry could be a PhD guide, and in collaboration with university, like a Symbiosis, we can have more PhDs. Now I work with persistent, and then the persistent, we have a lab where we have PhD guides available, and therefore in combination of PhD is created with persistent and Pune University. I think same model can be converted further. And therefore what is required is going and creating center of excellence. Now I can quote and some example of Sweden, where Vinnova, the Swedish government agency, has 19 excellent centers. And therefore these are some of the examples of the Kista Science City. I'm not going to go in much detail because I'm just trying to put some examples where universities can play a better role. I am very happy, and I thank Dada, Dr. Muzumdar, for getting a lead, that I have seen first time so many centers of excellence doing research in symbiosis. It is all aspect of life. It is in stem cell, nanosciences, medical imaging, AI, and I think this is one step ahead. So why I appreciate at the same time, we need to go beyond that. A university should just not remain what is called as an output giving, say, learned and expert software, expert manpower, but it should become a business engine. And therefore, once you do that, we get a lot of funding available by creating these innovation centers. There may be startups, and therefore we need to think that we become a nodal point for encouraging startups under the leadership of university, where they can be given in campuses that some of the areas we are already doing, but I need to widen that scope. I can give an example of in John Hopkins, where the nanomedicine center, it gets more than $10 million. I think now we have also a nanotechnology center. I think we are putting our foot forward, but then we need to amplify all these reports. And therefore, what I want, because Dada sir is here, that we need to think beyond that. Symbiosis has to become an umbrella for giving all those scopes to people and the creating a healthcare cluster, because the 28% revenue of persistent comes from healthcare. So healthcare is one of the growing area. And I think we need to give a lot of scopes that healthcare clusters, healthcare startups, medical instrument equipment engineering, all these need to become in a say affordable fashion. So with this, I just want to say that Symbiosis SIU should become a one-stop shop for technology transfer, RD publication, technology consultancy, prototype development, commercialization of innovation, patent and technology information, reverse engineering. All these things should go to all the industry. I think this is under inspiration of Professor Muzumdar. I think SIU should take this effective step to become in a golden jubilee year, one of the most progressive university across the country. We have a lot of problems in India because issues in research are India, who asks for it, who needs it, who wants it, is it demand driven, it is market driven, is it need driven? I think ours is, we are not need based and not focused on important areas, and main thing is quality. And the quality of research has to be improved, and we are very peculiar people. That now this slide shows you that whenever Indian scientists are in collaboration with Western scientists, the quality of research goes up. So why not ourselves think that we are best? And then I think this is the one which I want to put in, that the quality of the research has to be improved. So as a Vice Chancellor of MEHS, I did a lot of, say, new things. We permitted a cross-faculty PhD guide, interdisciplinary PhD, and then we put a new idea, which was coming from Knowledge Commission, that if a PhD student, after registration, has 5 papers of high-quality international journals published, he need not submit thesis. I think this is one of very progressive thing, which is taken up by MEHS, but subsequently they didn't like to say pursue further. I think SIU can think of that. And then we need to find out that we have all those labs and industries coming with the collaboration with, say, Symbiosis. We can have the PhD guides and co-guides. And the last important thing, which I want everybody to follow, is research methodology is an ignorant area. We should have a course for PhD students and guide, at least for 15 days before they land up doing any PhD work. So this is my brief introduction of philosophy of whole thing that is going towards research as a culture in SIU. And I appreciate all the efforts. At the same time, these are my thoughts. Now let us go down to what is the research process. Now, research process starts with aim is socially useful innovation. So problem formulation, initiation, solving, presenting, writing, and at the end, the research progress becomes the outcome, paper, patent, market. So this is a research process. And then it goes into these phases: define research question, conduct literature review, define questions, operationalize variable, design study. So that are the steps. So what I'm going to do is all the time, I'm just going to talk of all these steps, and wherever concerned, I'm just going to show you and give you where I failed and how I learned those processes. So I think failure teaches you better. Now, whole thing, a research question is most important. The research question starts from why. We need to open. We see a lot of new things. So why? Now, the slide is showing you an aircraft on the right side, and down is called ESWL, extracorporeal short wave lithotripsy. Now, this is a question to everybody of you. What is the connection between these two? Just put on chat box. Thirty seconds. Okay. The aircraft was manufactured by Dornier. When a Dornier aircraft was taking a takeoff, one ground engineer observed that there are fractures on the metallic debris on the airport. So this was a cause and effect relationship. Fracture of the metallic debris and the takeoff of aircraft. He thought there is something with that aircraft takeoff. Therefore, they went up and found out that there are waves. So these waves were created because of takeoff, and those waves were studied. And they were further modified. And through this simple observation of a cause and effect relationship, the biggest equipment, which is most human to the management of kidney stones, was discovered. That's called extracorporeal shock wave lithotripsy, which is, you have to put, it's an external source that you do not do any operation. You have to put it outside, and the stones, kidney stone, pancreatic stones, can be crushed. So a simple observation has led to the biggest discovery. Second, cisplatinum. I hope you all know cisplatinum is an anti-cancer drug. The scientists were studying the migration of cells across the cathodes to anode under microcurrent. The electrodes were made up of platinum. So somebody observed that the cells do not grow around electrode. Why? And then that question was given answer. They just took that fluid out, and it was a compound called cisplatinum. And the cisplatinum was just discovered in this small observation, which is one of the very nice anti-malignant drug for head and neck tumors and many tumors. So I think this is what is important, is every research question starts from why. So it's a uncertainty about health issues. So why? And then why research question? The well-crafted research question guides the systematic planning of research, formulating your questions, precisely enables you to design study, and we have a good chance to answer them. So I think basic aim, the basic thing of all research is a research question. So what could be research question? Now, your new ideas. You know the story about Fleming Flory, where in culture was left open. Fleming went on vacation. He came back and he found out a mold growing that inhibited growth of the staphylococci. Now that whole thing was identified as a Penicillium notatum, simmered the mold and extracted the antibiotic. And therefore, through this small observation that there is no the growth around of Staphylococcus around the mold. The 1938 mold was taken by Florian Chen, and 1945, the first antibiotic in the world was discovered, and they were given Nobel Prize. So the research question is, can the mold which killed Staphylococci in culture be best used to treat patients with staphylococcal infection? I think it's simple. Then Waxman, typical bacilli destroyed in soil, actinomyces. So he thought, why not have some similar antibiotic like penicillin? And therefore he isolated streptomycin. And therefore he was awarded Nobel Prize in 1952. So a soil organism similar to the penicillin from which antimicrobials can be produced. I think this is one thing. These are the things. What is important is to develop all these research questions. We have to challenge existing dogma. Do we need a mechanical bowel preparation for colon? Mine will be all medicine. Then identify importance to the India, and therefore intellectual intuition has to be there. And therefore we need to find out what are the priorities of India, like infectious disease, tuberculosis. Now BCG is coming into picture, and we're getting protected against viral epidemic. So I think vaccines, malaria. So these are the priorities. And as SIU, we need to find out what are our problems of the society in that area. Maybe, and those areas, maybe epidemiology, what are the healthcare problems, need to find out. And then subsequently we go to the district or maybe state and upward. That becomes our priority. I think we need to promote undergraduate research because they have sparks. You can see the medical college in Lavale, we're getting top class students. And therefore we need to promote undergraduate research from beginning. And now I can give you several examples of undergraduate research. Heparin, insulin, cyanoarterial node, ether anesthesia, they were all discovered by undergraduates. Heparin by MacLean, the insulin, which was given by, say, Best, who was a student of Banting, and ether by Valerius. Not only that, one of the PhD project, which was in the, say, Bergensteins and technology, where they were trying to see artificially generated stem cells. And therefore, through a project of undergraduate, the potential cancer management was there. Lanax was discovered as a postgraduate project, and then subsequently it is open to the public. So I think these are some examples where research has to become habit, skills, desire, and knowledge, and both things has to be put together. So I think we need to understand how the research can go. Now, I'm giving my own example that while I was working on xenogenic transfer factor, we need to find out, we have to concentrate that solution under 40 degrees of temperature. So we required a freezing centrifuge. So it was very expensive. So I told to my boss, Dr. Gharpure, that I can do by putting that centrifuge in the freezer and do same thing. And therefore the invention becomes a mother of necessity. I think that whole thing was there just because for the innovation. Now, one of my PhD student, he has Dr. Hemant Bansali. He has 9 patents, and one of the patent is on CO2 insufflator, which is volume specific. In a laparoscopy, you have to put the gas inside, and then there is a pressure sensitive. So instead of the pressure, because what is insert, what is required is the volume. Therefore, he created a new type of insufflator, which is volume specific. So I think I just want all of you to just give one research question. I don't know how can. Can you put?
Yes, sir. They
Put,
can put it in chat box.
okay, but how to? Because once I'm sharing the screen, I'm not able to see anything down.
So we will come to know. Adori, please help him.
So I'll dictate it from the chat box.
Okay, just put some research question, because that is something which I want people to get involved. Anything written, anybody?
So we have a question, a research question. It says, how can banking industry involve competitiveness for highlighting financial inclusion?
Thank you. Anyway, I think I have a bias of medicine, so I may not be able to comment on non-medical. So I just want all of you to give a research question and send it to Dr. Sharvari Shukla so that we can go ahead and refine.
Definitely, sir.
And obviously, everybody is aware about who gives money, and so the various agencies which you can see on the slide. And believe me, if you have a good problem, money always follows. Obviously, we require an ecosystem like an SIU. Now, this slide shows you how to kill good ideas. You go to anybody with good ideas, he says it's against company policy. It doesn't fit in the system. It will never get approved. So I think we have to appreciate whole ecosystem within the symbiosis, that we will never kill your good ideas. Now, I'm not going to much detail about how to form those questions, but then this is just slides for what is called as self-directed learning. So these are the what's called as ask, identify, choose, assign, manipulate, measure, and analyze. I think this I want students to go through and create a process by which they can have. So with this background, now you have a research question. Now, the moment you have research question, you want to just find out whether that research question has been already known, because that's what's called the research. Research means it's already searched when researching again. So what is important is do a review of literature. Now my all review of literature is pertaining to medicine, but then you have to start with textbooks, maybe latest editions, journals, NIC, which are various websites, and then various databases. And these databases are medical, but then I think everywhere databases are there. And then basic knowledge and whole thing has to be done. And before you go to do actual research, you do a review and to find out what is the exact level of knowledge. These are the various websites. These are all medical, but I think you need to extrapolate. And therefore you have to look for conceptualization, how variables are defined, measured, sample size, study design. I think this gives you clue. Now I'll give a classical example. Now I work on the immunosensitized pig cells, and then my boss got transferred. The next boss came. He said, you work on regional cyclophosphamide chemotherapy. Now, cyclophosphamide is an anti-malignant drug, and he thought we should give it through artery because I was working on maxilla. So give to the internal, say, external carotid to the maxilla. And then I did 14 cases of intra-arterial cyclophosphamide, but it's called regional chemotherapy. 14 patients, nothing happened, except 2 patients, their lymph node size was reduced. None of the patients, there was any additional, what you call as benefit. Now, we didn't do any review because my boss told me to do that. I am an obedient MS student. So I did that and then realized once we are doing to do review that cyclophosphamide for getting activation requires hepatic circulation. Therefore, there is no use in giving cyclophosphamide to the artery because at then it has to go to the liver and then get metabolized. Therefore, that whole thing was absolutely irrelevant. And believe me, it was a negative paper. No effect at all. And out of all my that immunosensitized porcine and lymph node cell work, this was the first paper accepted because it was negative. So even negative results are equally important. So the two things, do review first, and second, negative results are equally important. So I think with this, we have to go down and then refine the question with the knowledge. And if that question is, it has to be clearly stated as a question, testable, define variables, define sample size, samples, and setting. Now I play tennis. I like to quote something from tennis. We want to compare. Can you compare Federer and Nadal? No. Can you compare them as a tennis players? No. Tennis player in Grand Slams, tennis player in French Open, and tennis player during 2005 to 2010. So I think how we are refining that question. So the right title, let us say, I can extrapolate this. Can you compare ondansetron and metoclopramide? So if you go ahead, it has to be a randomized controlled trial for the comparison of milligrams of ondansetron and milligrams of metoclopramide in postoperative relief of nausea and vomiting for 24 hours using nausea scale in patient undergoing spinal anesthesia. So this becomes a research question, which is refined. So you have to find out what is type of study, say groups of subgroup study, dose response relationship, time response relationship, and outcome. This whole thing has to come into a research question. Then and then only you can have a better research available. So randomize the type of study, anesthesia, the group, dose, duration, time, say what are the outputs, and therefore this becomes a complete descriptive research question. Now we have to design a research study. So the three basic measure question: what do you measure? How do you measure? How will you measure? I think this whole thing is given as a basic sciences for statistics. So medically, we have incidence rate for the new cases and the prevalence rate for the total cases. Then I think rates, ratios, proportions. So we have usually two types of studies: descriptive study, what is called as a cross-section study. A cross-section study is a single point study which gives a prevalence, one time, no follow-up, easy, less time consuming, low cost. I think this is usually we do a cross-section study. And then we have a longitudinal study where we need to follow, and it is incidence rate, multiple visits, it's costly, time consuming, but then it gives you a better picture. Now, I just want to describe my experience of what is called the immunotherapy of cancer. Now, this whole project was done in Government Medical College, Aurangabad. Now, the process is I work on carcinoma maxilla. So take a tumor of carcinoma maxilla, cut into pieces, open a pig, that is the dirty pig. So you require an animal house, animal theater, open his abdomen, and in mesentery, that is the what is called as epithelium of the bowel, you create multiple small pockets and put that tumor inside. Close the pig. 10 days later, open it. Now the tumor inside that pig mesentery starts to grow. And then its growth is stopped by the lymph node cells. And therefore lymph nodes, which are very small, they become big in size. Usually they might weigh maybe 10, 15 grams. So they become almost half a kg. So these are the lymph node cells which have direct inhibitory effect on the tumor. And therefore we remove those cells and we need to deliver lye. So we created what is called as a solution in a tissue culture medium and then give intra-arterially so that it reaches directly to the tumor. So that if it goes to the body, then we have a problem of rejection. So this was my, say, research project for MS General Surgery dissertation, and which was done in a Government Medical College, Aurangabad. So you can imagine that a small medical college can offer you this much ecosystem. Now, the first time which we tried to do, the problem was getting pig. So in a government system, the dean of a medical college gave me grant and we purchased the pigs to the government money. Then we required an animal house, an animal theater. And nobody has given anesthesia to pigs. So we went to a veterinary surgeon. They say the only operation they do is castration. So when you are doing it, they only coming to watch. The first surgery of putting tumor in a pig, it was who's who. Professor of anesthesia giving anesthesia to the pig, professor of surgery operating, and whole animal husbandry department was in theater. So first thing, took the incision. My boss said, we open man, why you bother for pigs? He took a long incision and suddenly he found out the bladder was open because bladder in pig is intra-abdominal, whereas bladder in man is pelvic. Anyway, we took it that and then put it inside, and then suddenly the anesthesia was important. So they give, say, pentothal, and then we have to find out how the anesthesia has to be given. Say a pig has only two veins, two ear veins. And therefore the moment the pig was getting out, then we found out that there is no way solution available. So we put in pentothal sodium solution and give a peritoneal wash. So this was so much of experience to create a pig cell therapy. Then we took out those cells, we created a tissue culture medium, and then my professor was given me one, say, what do you call a lab technician and a whole one ward to create an immunology laboratory. And then subsequently, the first patient we gave, and then it was to be given intra-arterially. It is a foreign protein. The patient went into shock, and therefore we had to give adrenaline, and then with a titration method, we were able to find out and give that drug. The first patient, at the end of 1 week, the tumor started shrinking. At the end of 3 weeks, half of tumor was gone. And then we realized that it is very most effective. And then suddenly the basic defect, we have never taken a picture of that patient before therapy, because in those days, taking picture was very difficult. There was something called as a department of illustration with a photographer. He has to come. And then my boss got fed up. He gave me a brand new camera, which he got from US. He said, keep this camera with you and then don't ask again. So I think these are the mistakes which I did. Subsequently, I think I'm not going to much detail. And then we wanted to have the lot of sludge was there. So I did some 14 patients, and then 5 of them, they had complete remission. And I think this is one of the, what is called as very, say, original work in the area of what is called immuno-oncology. We wanted to, by the time everything was there, I thought I'm not going to pass because all my time was gone into doing surgery. And in examination of MS, I was going number 1, and all the time, that whole viva was on my immunotherapy. So I think then I requested my examiner, why don't you ask something about surgery? They said, you always know. And then we applied to ICMR grant, and we got a grant of some 60 lakh rupees in those days. But subsequently I passed. I was shifted to Ambezogai as lecturer in surgery. And then that whole project was out. So I thought, let us do something. And then I tried to do immune RNA. And I thought instead of say putting a project on porcine lymph node cells, we should put a project on immune RNA. But then that whole thing was gone because there was already paper available. So because people are smarter. So with this thing, I just want to go ahead. Now, research studies, we have 2 types, observational and experimental. We have descriptive, then analytical, case control, cohort, then experimental, we have RCT and non-RCT. So I think analytical study with the cause of effect relationship, So it could be prospective or retrospective. And this I'm giving you to give only basics, which is not there, and combine. So occurrence of event, you can go behind, that is retrospective, and ahead, that is prospective. And these both the studies are there. So let us say there are two basic types called as case control study and cohort study. The case control study is retrospective. Now, suppose we have a BCG vaccine given. So just find out people. Now, the study is efficacy of BCG vaccine in prevention of clinical leprosy. So take patients of clinical leprosy and take equal number of patients which are normal, which are controlled, and find out how many of them are given BCG, how many are not given BCG. And then we found out what is called as odds ratio. And then this is a slide which shows you that BCG is protecting leprosy by 10 times because the odds ratio is 0.1. So by and large, the studies are done retrospective. This is easy. And then we do a lot many times. But then what is important, we require a prospective study. It's called as a cohort study. So let us say effect of fetus on exposure of radiation in the first trimester of pregnancy in primary Gowra patients. Here also same thing, but it is prospective. So the cohorts that is exposed and control unexposed, find out how many of disease, how many of number of no disease, and find out what is relative risk. So the cohort studies are better because they are prospective. We need to follow them further. So we have to have a study design. I'm just going to give you about clinical trials because these are historical.
लेकिन अभी हॉस्पिटल में भी है हॉस्पिटल में भी
So core components is all human subjects. Most of them as control group, most of methods were measure. So I think I must go into details about the common thing, what is called as a randomized double-blind clinical control trial. Now we do what is called the triple blind, where even the computer guy who handles also doesn't know. This is called triple blind. And then randomized scheme to assign participate to one group because we don't know which patient belongs. So neither patient knows who has given drug or which is the placebo. And I think this is what is called as today's standard investigation 2 or more conditions, and then inclusion criteria, exclusion criteria, the sample size, then half of the people, they are not given drug, that is placebo. And sometimes even the placebo works because there is no drug, because placebo is something which has no drug. But then sometimes just because of the process is there, he might get benefit. One of the biggest thing that has happened in carcinoma prostate, where there was total androgen blockade, and a patient with total androgen blockade, the placebo was giving better results. So these are the phases. Phase 0, the pharmacokinetics animal trials, and then phase 1, because everybody is aware about the phase 1 trials and phase 2 trials in say COVID vaccine. Phase 2 are multicentric, and then phase 4 are community trial. So phase 1, the small groups, and phase 2, the large groups, and phase 3, more around 3,000 people, because at the same time, a COVID trial is there. And I think I'm not going to much detail about all these trials, but these are the study designs. We can have concurrence, initiation of study, end of study, study group, control group, and then in a crossover, midway, the study group is converted into control group, and then it's a crossover. So these are the various trials which I conducted, and most expensive was we were first to conduct a clinical trial of Viagra, that is sildenafil citrate, in male erectile dysfunction. That give a far more experience. And I think I'm not much going to detail of these because these are all trials. So wherever you want to go trial or any study, what is most important is the sample size. And I think statistician plays a major role because unless we have a effective sample size, all research becomes irrelevant. So that's why the specialist job is done and we require a specialist. So a statistician has to be a part of research team, and therefore we need to find out how best we can use for creating a design. And the services of statistician are must for all research projects. And the other thing which are coming latestly is what is called as umbrella clinical trials and basket clinical trials. So in an umbrella clinical trial, what is called as one disease with multiple types, either because of say clinical types or the next generation sequencing, and multiple drugs are given for the same drug, same disease. So in umbrella, one type of cancer and multiple genetic mutations and multiple drugs simultaneously. Whereas in a basket clinical trial, we have one drug which is useful in multiple cancers. So this is say umbrella, where drug A, drug B, drug C, depending on the mutation. And we have everywhere controls. So I think this is coming in big way, what's called as umbrella clinical trials. Whereas a basket clinical trial, single drug, like say EGFR, and where multiple say diseases are there, it could be useful in lung cancer, colorectal cancer, and head and neck cancer. So single drug in multiple cancers is what is a basket clinical trial. These are the two things which are coming in big way. So obviously we require ethical permission, and we have ethical committee everywhere. And in SIU, in medical college, we have two different teams, one for the clinical trial, and we have to find out how we handle ethics. The three ethical principles: respect for the person, treatment of person has to be autonomous, no conflict, and no damage. And we have to give a treatment of all fairly. So ethical norms, the randomization, proper use of placebo, process of majorities, let's say competent investigation, informed consent. Consent has to be detailed, telling you everything. And usually we have what is called as scientific advisory committee, community advisory committee, to find out what are the factors. So these are the clinical trials. So with this background, as I earlier suggested early, we should have a technical committee for research for all institutes of SIU, where expert in research who has fascination should be on the committee. And it should not be by hierarchy, that those who are interested in research should be taken on the committee. And statistics has to be compulsory. The research papers are rejected more often by research design, and the agencies even granting permission for the budget, they also look for research, say, guidance given by a statistician. And medical colleges need to have, in addition to clinical trial, drug trial, medical committee, and the MHS has already done that. They want one technical committee to take care of, say, research project across. Then analyzing data, I think this whole thing, I'm just going to flash and put it for what you called as self-directed learning. So this is whole thing is known. I'm just going to flash those slides. What is mean and what is standard deviation and statistical test, probability. And I think this whole thing is statistics. So this becomes a part and parcel for any research. So once you have data available, you need to publish. Because if you want to finish, why did you start? And if you don't want to publish, why did you finish? Therefore, you need to finish and publish. And then you have to either present in conferences, usually start with an, say, respective specialties from city, state, and Asia Pacific, those things are available. And then you can publish in respective journals. I'm giving you a set of examples of medical education. I think we need to find out good journals where we can publish. So my project for PhD was on NK cell interferon system. So it's a natural killer cell interferon activity where it has all those things available, and interferon was found to be useful in oral cancer. And therefore we did a lot of things, and everything that we did, we had 6 PhD students working under Dr. Ghosh, and we did everything and we published. The first paper that was very important was that when these cells are cryopreserved and then they are heated, the interferon producing capacity and other interferon parameters are preserved. So this was the paper which we published in 1989. I think this was the basic paper on which all cryobiology is dependent, IVF and whole technology dependent. But I think that whole thing was done by my teacher in 1989. So then we need to apply for patent. So I don't want to go in much details, but I think a statistical institute like SIU should have a separate area, and maybe we start a course in patent. I just want to end with a review process that once you submit, what happens? Now, these are the 2 resources, one from BJU, where BMJ has given the resources for author, where they have given in detail about how the papers are reviewed. And this is an equator network where resources of good reporting. I think we have to go into detail about how papers are reviewed, and we have to prepare them accordingly. An International Committee of Medical Journal Editors. So that these 3 references are very important to find out how the papers are evaluated. Now, 9 reasons for acceptance: relevant problem, well written, well designed, thoughtful. Now, well written, most important. I passed my MS in Aurangabad, and I was appointed as a lecturer in Ambezogai. And then I first sent my manuscript to Indian Journal of Surgery. Those days, even manuscript writing was with a typewriter, and so very tedious procedure. I got a letter from Indian Journal of Cancer that it is accepted. And suddenly, after 1 month, that whole paper was sent to me under a pretext that it was not well written. Rather, the language was poor. Now, I am trained in a Marathi medium, and therefore I think in Marathi and translate in English. Therefore, that whole paper was written by us, because I was in Ambezogai, the guide was in Aurangabad. It was badly written. Then I took a chance. I went to a professor of English in Ambezogai and asked him guidance to find out how to write an English paper. He guided me, and subsequently I submitted that paper to Indian Journal of ENT. So what is important? You require a good English. So I think that is one thing which I learned. It has to be literature review, the sufficient sample, practical implication, acknowledgement. So these are the reasons of acceptance. And then 10 reasons for rejection. First reason, inappropriate statistics, overinterpretation, inappropriate suboptimal instrumentation, sample size too small. That's why we have to think from sample size from beginning. Text difficult to follow, writing, inadequate problem statement, inaccurate data, inadequate literature review, insufficient data. So these are the causes for rejection. So I think while you are submitting a manuscript, we have to find out these reasons, and therefore the study design has a strength, data analysis has strength, and therefore all the time I'm coming back to statistics, that study design has to be proper statement, conceptual record, research question,
So sorry to
the
interrupt.
relevance.
So sorry to interrupt.
Yeah.
So how much more time will you need?
2 minutes.
2 minutes. Okay, thank you.
And so I think this whole thing pertains to statistics, that all the paper while you submit, you have to have proper study design and proper data analysis, and manuscript writing has to be presentation of results, discussion. Now, while you write, you have to go back and find out the particulars of each journal, because every journal wants separately. Each journals want bibliography separately. So I think we have to study, and depending on the requirement of individual journal, we have to submit. So these are the practice. So I think with this, I just want to say that publication is equally important. And why publish? You have to find out a good journal, but that good journal will accept good article, provided it has good design and good manuscript and good content. And therefore it will accept. And therefore, I say that we need to create an academic leadership, especially in SIU, to find out how better we do research. So leadership is about creating an alignment of strength and making people's weakness irrelevant. So thank you, Dr. Sharwari Madam, for giving me opportunity to talk with rather non-medicos. And I just try to give all research methodology the new angle, where nobody will say that these are our mistakes, because I learned from mistakes, and that might be costly, but that I learned. And I don't want people to follow my mistakes and study and do a better research. Thank you.
Thanks indeed, sir, for joining with us, during odd hours sitting in US. Thanks. Thank you so much.